Okay, welcome back. This is Issues in Biotechnology, the way we work with life. This is Applications in Biotechnology, and we're into the section on medical biotechnology. We have already looked at medical research history from the development of prostheses through stem cell therapy and including animal and human cloning and genetic engineering. We then looked at gene therapies, tissue engineering, and how nanotechnology will interface with medical biotechnology in the near future. In the last set of lectures, we looked at pandemic flu and bioweapons. Tonight, we will look at the biology of cancer and emergent technologies. So pandemic flu and bioweapons was an analysis of how current DNA-based technologies will influence our ability to study and perhaps control the next pandemic flu. We also saw that this creates a dual use of knowledge dilemma. Just a reminder that the final biotechnology stocks project is due by the completion of this course. So if you're out there online, you might want to check in on those numbers. So cancer biology, what is it? How does it happen? And what would be the emergent treatments that will evolve out of our now knowledge about genomics and DNA? Cancer. That's a tough word. If you hear it related to yourself, certainly it has more gravity than just this slide. If you hear it related to someone you know, it also has a deep emotional impact. Certainly the word itself carries a lot of gravity in our society today for a lot of reasons. Do you know someone who's been affected by cancer? Usually, most everyone in the room would say yes. Do you know someone who's been affected by cancer and is a relative? Usually this is also a very high percentage of people. This underscores the genetics and the biology component to cancer, but as we will discuss, there is an environmental by genotype influence of these diseases. If you were just diagnosed with terminal inoperable cancer with only a limited time to live, you would most likely A, seek the best possible treatments, B, spend as much time as possible with people that you love, C, seek solace in your religion, D, live each day to its optimum, E, seek a convenient end. These are all choices that probably anyone who receives such a diagnosis experiences. So let's look first at the biology of cancer. What are some cancer basics so that we can get our head around this as a biological phenomenon before we look at how we can mitigate or intervene in that biology. So in the first part, we will examine first the fundamentals of oncology, that is the study of cancer. What are the socio-economic burdens that these diseases bring to our society? What is the origin of cancer, or what we call carcinogenesis? And then how does this present in terms of its pathophysiology and its molecular pathology. Understanding these bases for its biology will help us then examine what could be the possible treatments based on targets, specific biological targets. And we will look at a series of contemporary targets as they may play out as case scenarios for future research. In the second part, we'll look at the applications of biotechnology in cancer treatment. I'd like to discuss a brief history of cancer chemotherapy, where it came from, how it's applied, and then look at small molecule development as it now is playing out post-human genome, and then give 3 case studies on new drugs that are making a difference now so that we can understand how current research might play out in the future. The course on biotechnology that we present here is meant as an overview. Certainly, some of the topics that we cover could be entire upper level, if not graduate courses all unto themselves, and certainly cancer is one of these. So what we hope to accomplish in this lecture is to provide an overview so that you can get deeper into this topic if you're interested. And there are many good textbooks. This one by Robert Weinberg on cancer principles is one such resource, but actually there are many. Cancer is known medically as malignant neoplasm and is a broad group of various diseases, not just one, all involving unregulated cell growth. So cancer is not really just one disease, but a group of diseases that are characterized by unregulated cell division. So cancer then is the uncontrolled growth of abnormal cells in the body. Cancerous cells are also called malignant cells. Cancers in general are also caused by a series of genetic mutations. Each mutation in a series alters the behavior of that cell somewhat. As a hereditary component, you may be born with already a certain subset of these mutations, whereby exposure to certain environmental cues, you could acquire then the requisite next or subsequent mutations required to trigger unregulated cell growth. Cancer is also heterogeneous. Most cancers have not one type of mutation, but hundreds. Cancer grows out of normal cells in the body. It usually originates from a single cell. Some people find this difficult to comprehend, and I'll get into this phenomenon later on in the lecture. Normal cells multiply when the body needs them and die through a phenomenon called apoptosis, or programmed cell death, when the body doesn't need them. Cancer cells acquire characteristics to evade apoptosis. Cancer occurs when the growth of cells in the body becomes out of control and the cells divide very quickly. It can also occur when cells forget how to die. So the type of cancer, so-called, is usually described by its organ of origin, so that you can see in common literature lung cancer, prostate cancer, breast cancer, colon cancer, etc. So we rather crudely characterize these diseases according to where they start. This is a image of a CT scan showing lung cancer. This is a photograph of an invasive ductal carcinoma of the breast. The yellow area at the center is the tumor, surrounded by spikes of whitish scar tissue and yellow fatty tissue. This is an invasive colorectal carcinoma and a colorectomy specimen. This is a squamous cell carcinoma, the whitish tumor near the bronchi in a lung specimen. So you can see that different tumors present different phenotypes and in a multiplicity of different organs. Malignant tumors can invade nearby parts of the body or nearby parts of the organ in which it has originated. The malignant cancer can also spread to more distant parts of the body through the lymphatic system or in the bloodstream in a process called metastasis. Not all tumors are cancerous. Benign tumors do not grow uncontrollably as do malignant tumors, and they do not invade neighboring tissues and do not spread through the body through metastasis. There are over 200 different known cancers that have been characterized that afflict humans. Although humans are not the only animal which experiences cancer. And since 1971, people talk about the war on cancer, or finding a cure for cancer might win you the Nobel Prize, as if it were just one disease, or as if finding a cure would be such an epiphany. The symptoms of cancer spread, metastasis, depend on the location of the tumor of origin, for one, how rapidly it's growing, and its molecular biology. And people say, what causes cancer? I've already alluded to the fact that it has a genetic and biological component. But there are certainly stimuli known as carcinogens, which can influence mutations in the series of genes that could lead to uncontrolled cell division. These would include aromatic compounds like benzene and other chemicals, drinking excess alcohol over a long period of time, smoke inhalation, particularly tobacco. There are a series of carcinogens in tobacco smoke, but the nanoparticles that make up tobacco smoke also may stimulate cancer development. We are bombarded by environmental toxins, and certainly each of our independent genetics renders us somewhat uniquely sensitive to each one of these, and they're all over the place at increasing levels. Some of these are hormone mimics, and hormones themselves, whether endogenous or exogenous, can play a role in the development of cancer. Excessive sunlight exposure and UVB radiation can play a role in making thymidine dimers, where two T's in DNA become dimerized, and this has been shown to influence the development of cancer when it occurs in certain genes. I've already mentioned the component of heredity. Lack of exercise has been shown also to play a role, as has obesity. Although those might be related in some cases, the role of exercise in autophagy of cells, that is increasing turnover of degrading enzymes and cellular components, seems to also play a role in the decreased risk of cancer. Exposure to radiation. whether that is from X-rays, flying in airplanes, radon gas, other environmental exposures, or catastrophic events that may occur as in a nuclear reactor failure or an atomic explosion. Viruses are known to play a role in the development of cancer. Hepatitis B is one of the leading causes of liver cancer. Human papillomavirus has been demonstrated to be involved in cervical uterine cancer and esophageal cancer. Latent viruses within the genome have developed into so-called oncogenes. So the ancient viral infections that are now hitching a ride within humans that no longer stimulate viral replication can themselves play a role in the development of cancer. However, the causes of many cancers remains unknown. Here's a brief history. I like to set these into context in this course. How did we get here in our knowledge base? Well, in an examination of cancer, this actually goes back quite a ways to roughly about 3000 BC, when on papyrus scrolls we see a description of cancer of the breast. And Hippocrates coined the term cancer to describe tumors about 400 BC. John Hill described tobacco snuff as a probable cause of nose cancer in 1761. In 1846, anesthesia became available. This was obviously a benefit to surgery, but it had a large influence on the ability to surgically remove tumors, and so surgery became perhaps one of the first really beneficial treatments at that time for cancer. And then Wilhelm Röntgen invented X-ray technology, and radiation therapy of tumors followed closely. The mustard gas that was used in World War I was found to reduce white blood cells, and I'll describe more of that when we talk about the development of chemotherapy in terms of the so-called Bari myth. But chemotherapy was born in 1919, but really came to a forefront with the first remission of pediatric leukemia for the reduction of white blood cells in 1947. Then we enter into the advent of molecular biology with the description of DNA structure by Watson and Crick and all of the developments that followed into the later 1950s and 1960s. In 1964, the U.S. Surgeon General reported that there was an undeniable link between smoking and cancer. It would take decades before an anti-smoking act was signed into law in the United States. In 1971, President Nixon signed the National Cancer Act, and the war on cancer was actually declared and became public in the common literature such as Time and Life magazines. Computed tomography, or CT scanning, was first introduced in 1972, and that really revolutionized radiology and tumor diagnosis beyond biopsies. Janet Rowley, MD, showed in 1973 that chromosomal abnormalities were present in some cancer types, and in this case, specifically describing translocations between chromosome 9 and chromosome 22 in humans. This led to further descriptions about chromosomal abnormalities associated with cancer and tumor cell development, particularly aneuploidy. 1981, the FDA approved the first vaccine against hepatitis B. The 1990s saw overall cancer death rates begin to fall in the United States and Europe owing to chemotherapy, radiation therapy, and surgery. That triad of treatments remains constant today and has influenced an overall decrease in death rates and a more optimistic viewpoint with a cancer diagnosis. And I'll talk more about this later in terms of hope for future. And everyone always asks me, what's the worst kind of cancer? That'd be the one you have. Aside from that, there are treatments now that have been very effective at treating certain types of cancer. Gleevec was approved in 2001 as the first biotechnology drug to target a specific mutation. This is a small molecule drug, and I will talk about its introduction and its efficacy later. Now, cancer survivors have reached 12 million in the United States. Cancer survivors are designated as such 5 years into remission. That's a fourfold increase since 1971. Has cancer been cured? No, not quite. But effective treatments and our knowledge base has dramatically improved. This engraving shows two views of Dutch women who had a tumor removed, two views of a Dutch woman who had a tumor removed from her neck in 1689. Surgical treatment of cancers is still highly recommended in most cases. The socioeconomic burden of cancer is certainly a backdrop to this discussion. So let's look at this first. Cancer is, in our common language, probably owing to the fact that we all are familiar with someone who's been touched by this disease. We hear about it through Time, Business Week, Newsweek, and common literature. We get hopeful reports that there is new ammunition in the war against cancer. But let's set the stage here first. This is a view of the leading organs of origin sites of new cancer cases and deaths. Shown here for male and female, on the left for new cases, and on the right for estimated deaths by those cancer types according to their organ of origin site. On the left, you see that prostate cancer in males, obviously, accounts for about 33% of estimated new cases. In females, breast cancer accounts for nearly an equivalent at 32%. This area of the United States in the Northeast has a dramatically higher rate of breast cancer in comparison to the rest of the United States, and you may ask yourself, why? In both male and female, these rates are followed by lung and bronchial cancers at around 13 and 12% respectively, and then colon cancer at about 10 and 11%. We see that the estimated deaths, however, are at the highest for lung and bronchial cancer at around 31% in males and 27% in females. So these are typified by aggressive types of cancer that are difficult to treat by radiation therapy, surgery certainly, and available medications. This also shows the estimated new cancer cases and deaths as it occurred in 2005, and the number of cases in all sites over 1.3 million per year. This graph illustrates the age-adjusted cancer death rates for males between 1930 and 2001. There's a couple of things I'd like to point out in this graph. First of all, that lung and bronchial cancer increases dramatically between the years 1940 and 1990. This is directly correlated to tobacco use in the United States in men. So right around World War II, the availability of tobacco and its popularity and the knowledge that it was not dangerous contributed to this large increase. Secondly, I'd point out the drop in stomach cancer deaths from 1930 down to 2001 in this graph, and still steadily today. Why do we see a drop in stomach cancer? Except that it's operable. Actually, you can live without your stomach. So surgical removal of tumors and the availability of diagnosis have contributed to a fairly good survival rate for stomach cancers. Notice also that some cancers have remained relatively steady. Colon cancer, for example, but it has dropped off slightly since 1980 owing to the introduction of colonoscopies. Certainly, early diagnosis contributes to effective treatment. I will discuss colon cancer in terms of its staging and its diagnosis later on in the lecture, but also look at the drop in lung and bronchial cancer in men post-1990. That's about a 25-year lag after the Surgeon General noticed that there was an undeniable connection between smoking tobacco and cancer. And that's about the frame of time in which people have decided to curb those habits. So there is a 20-year lag time shown in this graph too between cigarette consumption in the United States and lung cancer, showing that the incidence of lung cancer in the United States, and now we see a similar type of an effect in China, is highly correlated with smoking tobacco. So the incidence of lung cancer is highly correlated with tobacco, and as I mentioned, it's more than just the particulate matter, but there are a number of carcinogens, some of them very potent, some of them less potent, but certainly at the dosage rate and what we will call field effect, treating a large number of cells within the epithelial of the lung simultaneously, can certainly increase the chances of mutation of a single cell. Let's look at that same data as it compares to women. We see a much lower rate of lung and bronchial cancer, especially in the 1930s, but we see a rise in lung and bronchial cancer in women after 1960. What do you think that's all about? Advertising, perhaps? Virginia Slims, You've Come a Long Way Baby ad. You know that now it's fashionable to smoke tobacco and represent your individuality. So the cost was paid in terms of health and life. The comparable drop in stomach cancer we also see in women. We see a decrease in breast cancer owing to probably the diagnosis capabilities of mammography, and also a decrease in colon and rectal cancer. Notice, though, that women have a lower incidence of cancer overall, and they tend to live longer. The cost of cancer to the United States is huge. Roughly about $190 billion total cost per year, divvied up among direct medical costs at about $70 billion. Direct morbidity, that would be loss of productivity. People can't work. People have to take care of people who can't work. And about $100 billion in indirect mortality cost. In the brief time that we've already been discussing this, about 200 more Americans will lose their fight with cancer. 1 per minute, every hour, accounting for about 1,500 more victims every day. The emotional impact is really unestimable. We can describe dollars, but this is really hard to quantify in any way. So the human cost is high. The emotional impact is great. 1,000 days survival from some drugs or the triad of treatment is really not enough. So I think there's a lot of hope that's presented by our current knowledge base about the biology of cancer and treatments that will come about because of the hard work of decades. To understand this, though, let's look at the origins of carcinogenesis. Cancer cells are released from normal cell division pathways and division restraints. Normal cells have a limited ability to replicate. Usually the subcultivations in cultured cells, the doubling will cease after about 30 or 40 rounds of subculture. This is owing to the biology behind programmed cell death and a number of other factors, including regulation of telomeres at chromosomal ends. Tumors are masses of cells that are derived from a single cell. So when a person develops lung cancer, it is not the entire lung confluently which develops a tumor. These usually have a single cell origin. If another tumor develops somewhere else, this could be because that tissue broke off, migrated, and invaded that tissue, or it could be an unrelated independent event. These can be genetically analyzed now, and I'll show you some examples later, but a series of genetic mutations accumulate in pathways that are involved with regulation of cell division, when to start dividing, when to stop dividing, and how cells die. There are also mutations that could accumulate that would influence tumor growth and intravasion of a tumor that spreads through blood vessels and the lymphatic system, resulting in metastasis. But it's a clonal disease, and this can be diagnosed through the technique I described in the first part of the class called Southern blot analysis. Now, even more precise molecular analysis shows that these mutations have arisen from a single cell. And then the expanding colonies of cells evolve with successive genetic changes which may initiate those other events that I just described. So the result of this clonal propagation in carcinogenesis is a clone of cells that is unresponsive to normal cell growth controls. And therefore, it's able to expand indefinitely and can be cultured indefinitely. I had mentioned previously human cell cultures called HeLa cells that were derived from an individual named Helen Lack. And these were sampled, I think, in the late 1950s and have been perpetuated in cell culture. You can order these now even from supply houses. And there's more of her around today than there ever was when she was alive. And actually, a lot of good cell biology and cancer work we owe to that individual. And she or her estate is not compensated. So even though it starts with a single cell, this is not as random or sudden as you might imagine. It simply does not come out of nowhere like a strike of lightning, but rather whole tissues become predisposed. So carcinogenesis is not random per se. Many cancers exhibit this field effect that I alluded to just a moment ago, suggesting that many normal appearing cells in a target tissue, say skin or lung tissue, may have elevated malignant potential. I already said that you could be predisposed by already having a series of mutations where exposure to UVB may trigger the last one, which would release those normal growing cells to become malignant. This is also consistent then with the molecular genetic view of monoclonal progression of cancer. However, it also implies that a field of cells with elevated malignant potential may exist that are clinically normal. Certainly this is true and would not be recognized morphologically or histochemically as being malignant. However, owing to genomics, pharmacogenomics, we can now analyze predispositions. There are certain target genes that have been recognized. BRCA1 and BRCA2 are great examples of genes which predispose individuals to breast cancer. These cells in a field effect cannot be detected as having higher malignant potential on simply a morphological or histological criteria. They look normal until downstream mutations are acquired. So in these field effects, you can imagine that the entire surface of the skin that's exposed to sun, epithelial in smokers' lungs, contralateral breast cancer, HPV infected uterine cervix. These are all examples of cells that are uniformly exposed in a field effect. So exposure to carcinogens is one of the aspects which triggers carcinogenesis. What is a carcinogen? A carcinogen is a compound or a component which may influence a mutation in a field effect. If this mutation is random, it may affect genes that are uninvolved in carcinogenesis. That will not necessarily involve cancer. Although the Ames test, so-called, has been used to analyze compounds according to their ability to influence mutagenesis. Some of these highly mutagenic compounds, just by comparison, will be carcinogenic. So we can look at carcinogens in terms of ionizing radiation, chemicals, hormones, viruses, etc. But in the Ames test, let me describe here a little bit about how this test is done so we can evaluate the results of this test. This test is done using Salmonella bacteria that are histidine dependent, so that they're requiring histidine in the media in order to grow. To do this test, a suspected mutagen is added to a sample, and the cells are then plated onto a medium lacking histidine. Therefore, only those cells which have acquired a mutation that affects their ability to be histidine dependent will survive. Few colonies survive in the presence of compounds tested that are not mutagenic, whereas that number increases proportionally to compounds that are mutagenic, and they're rated accordingly on the Ames test. So you can find all kinds of compounds on the Ames test which are mutagenic and naturally occurring. I mentioned in our discussion on agriculture that there are compounds in celery, for example, that are mutagenic. Bread crust is mutagenic. There are compounds in caramelized sugar, which are mutagenic, furfurans. Compounds that you may not normally associate with carcinogenesis per se could be carcinogenic. There was an issue of Science on the cover not too long ago that said, eat food and die. You know, we're exposed to all kinds of things. Teriyaki chicken, the compounds that are derived from pyrolysis products on charcoal and cooked beef and meat are carcinogenic. So we're exposed to carcinogens all of the time. The point is, some of them are more mutagenic than others. Some of them can be avoided. Can you avoid all of them? I wouldn't recommend it. So we also want to distinguish between mutations that occur in normal body cells, so-called somatic tissue, where a sector that is mutant and could be tumorigenic, in fact, will not transmit those mutations to offspring. However, if mutations arise in germ cell tissues, those that give rise to eggs and sperm, those mutations, in fact, could be heritable. Not that a person would develop a tumor necessarily in germline tissue, but as I mentioned, this is a sequence of mutations. People are predisposed by having already one or more of these mutations in the chain that will eventually erupt as a tumor cell. Tumors themselves are complex biosystems, not a homogeneous cell type at all, but they may be comprised of tumor cells, as shown here, as abnormally growing cells in this histological preparation, next to normal epithelial cells, and you can distinguish those by their appearance in this histological slide seen under a microscope, compared to lymphocytes and necrotic tissue in this biopsy post chemotherapy. All of these tissues, including blood vessels that invade tumor tissues during the process called angiogenesis, create a complex biosystem that also further encumbers how we treat these tissues. In other words, it's not just a cell, it's almost an organ unto itself. And they're really smart. I don't mean to be anthropocentric about that, but the molecular mechanisms of tumor biology are such that it makes it difficult to devise any single strategy that will treat this complex biosystem effectively. So here you can look at this field effect as demonstrated in this histological example of prostate cancer, where this locus described as D18S34 and D18S35 reflect a common, perhaps earlier change in prostate neoplasia. In other words, they may have had a similar origin. D8S254 as a later change, perhaps more proximal to malignant disease. Cancer cells are created when certain genes mutate. Oncogenes promote cell growth and cell division. What is an oncogene but a gene that's involved in tumor progression and tumor biology? Amongst these, there are genes that have evolved in humans as tumor suppressors, genes which actively suppress uncontrolled cell growth, such that when you have a mutation occur in a tumor suppressor gene, you are already predisposed. This could be before you acquire the other subsequent mutations or afterwards. Cellular genomes are known now to harbor proto-oncogenes, and that some of these may have been derived from ancient viral infections or ones that occurred during an individual's lifetime. This slide demonstrates the stepwise, in this case, ordered genetic changes in colon tumorigenesis, starting off on the left with a normal epithelial cell that becomes dysplasic after a mutation in a gene called APC. Later, subsequent mutations in RAS genes and then eventually in P53 lead to developed carcinoma, and then subsequent other changes would be involved in those carcinomas to render those cells capable of metastasis. So full-blown tumorigenesis in this individual case requires 4 to 7 events of independent mutations. These mutations do not necessarily have to happen at the same locus, however. Mutations which interrupt gene function throughout, say, 1,500 base pairs or so could result in tumorigenesis. So could mutations involved in regulatory aspects of that gene, whether that is in the promoter region or transcription factors which activate those promoters. I hope what I'm trying to say here is that there are a number of different genes that could be involved with leading to tumorigenesis, and it's not necessarily the same genes in every single case, or in even every single case in any specific organ. So the hallmarks of cancer really begin with this release of normal cell division patterns, and that the proteins that regulate cell division that are encoded by genes are ones that are involved with oncogenesis. So this disruption from the normal cell cycle is what's liberating these cells to become tumor cells and uncontrolled cell growth. So let's go back to general biology, realizing that the cell cycle shown here on the right, you can start at any place on this arbitrarily, but it's normally thought of in terms of interphase, followed by the stages of mitosis, but the also comprising, shown on the inner ring here, a phase of synthesis where DNA is synthesized, followed by a G2 phase, which is called GAP2, characterized by DNA repair, followed by mitosis into 2 daughter cells, shown there in the upper right, and then continuation on into G1. Some cells go into a resting phase called G0, which can remain latent in that phase for extended period of time before either continuing on into S phase or going into programmed cell death. So this is a normal cell cycle function which could continue in cell culture for 30 to 40 rounds of subculture normally until going through apoptosis or programmed cell death. Remember, stem cells in the earlier lecture are a subpopulation of cells which also have a rejuvenating capability and remaining in culture for extended period of time, and these share this characteristic then with cancer cells. Remember also in that lecture I described induced pluripotent cells, where researchers were able to use clone genes that resemble cancer cells to reprogram normal adult cells to have the rejuvenating capabilities of stem cells, which then were released from that rejuvenating tendency can then differentiate into normal cells. So you can see here a parallel somewhat between stem cell development and cancer cell development. Not that one causes the other necessarily, but they have a similar biology related to control of cell division. So what are the key genetic changes that confer these advantages to tumor cells? Well, self-sufficiency to growth signals, particularly hormones, insensitivity to anti-growth signals in the cell or tissue invasion of a developing tumor and then subsequent metastasis, limitless replication, as demonstrated certainly by HeLa cell cultures, a sustained angiogenesis, which is fed by signals out of a tumor, which cause blood cells then to grow into a tumor and further accelerate its growth. I'll talk more about that later in terms of drugs that can be used to treat angiogenesis and therefore limit tumor growth. And then the evasion of apoptosis. Here are some of the properties of normal versus cancerous fibroblasts in culture. And then one of them that was recognized rather early is that contact inhibition to growth is absent in cancer cells, that these normal fibroblasts would grow attached to a substrate, whereas cancer cells have this property of balling up. And the rest of these growth factor requirements are high in normal cells that require exogenous growth factors, whereas low in cancer cells because they have their own endogenous factors or have receptors that are unresponsive to normal growth factor treatments. Like the contact inhibition of growth, normal cells have an anchorage dependency for their division and growth, whereas that's released in cancer cells. Cell cycle checkpoints are intact in normal cells and released in cancer cells, and the chromosomal profile of its karyotype is consistent in normal cells, but we see in cancer cells sometimes the translocation of chromosomal arms or whole-scale aneuploidy. One of the other hallmarks of genetic changes that occurs during tumor cell development is the evasion of apoptosis. This isn't merely just a simple change that results in the release of programmed cell death. I wanted to show this slide as an overview of the signal transduction pathway that's involved in programmed cell death, just to demonstrate to you that it's a complicated pathway from signal recognition down to actually gene regulation, shown down in the lower right of that enclosed box. So all of these different checkpoints represent proteins that are encoded for by genes. Any of these going awry contribute to the problem. You see the picture's starting to get dense, right? So there have been, I really should correct this slide. There have been 290 described genes that have been linked to cancer by identification of mutation. Actually, that list is growing every day, and you could probably update it every day based on new and current genomic analysis. These would include point mutations, where a single base pair changes, and I've shown you the consequences of doing that could result in frame shift, missense mutations, nonsense mutations, and so on, that would either inactivate genes like p53 that are present in over 50% of cancers, or activation of other genes that are involved in cell divisions like the RAS genes. Translocations can also occur, as I mentioned, whether that is whole chromosomal translocations or specific gene segments that can be translocated and either inserted or deleted. So off to the upper left of this slide is a list of some of the mutations which could give rise to the abnormalities that could cause release of cells from a normal cell division pattern. This is a demonstration of the sequence involved in the VSRC gene involved in some carcinomas. So the list of genes that are mutated somatically but not inherited in a mutant form that contribute to cancers is quite long. This is only a partial list, characterized by some of the somatic mutations and the proteins that they encode and the site of origin. So we don't really need to go into this. I really just wanted to show you that there are a number of genes that regulate checkpoints either in the cell division pathway or in signal transduction pathways that are involved in triggering release. So these sets of genes that I showed you just here, and this is really only a partial list, are involved in Go functions, start dividing. There are a whole nother set of genes that are demonstrated to predispose people to cancer that are involved in don't stop. So you can already tell by this list that if you put Go next to don't stop, you already have a problem that's accelerating. Now if you add in mutations in genes that normally suppress this, tumor suppression genes, you could have mutations in Go genes, and then tumor suppressor genes would act on those. You could have mutations in genes that regulate the don't stop functions, and these also will not necessarily result in carcinogenesis. So in other words, you have to have a lot go wrong simultaneously, but look at how often it occurs. Aneuploidy is particularly high in tumor cells, so that detection of defects by karyotyping has become part of tumor cell biopsies. So abnormal chromosome numbers of either whole chromosome sets or individual chromosomes are a hallmark of cancer types. Nonetheless, even though it seems as though this paints a very complicated picture, and it does, owing to the fact there are many genes involved in Go, don't stop functions and tumor suppression, the fundamental concept remains the same, that it's a release from cell cycle that's normal, and that there are other mutations that provide advantages such as increased growth, increased survival, increased angiogenesis, and release from apoptosis. Think about the biology that's presented here and its complexity, and how then, when we think about intervention, drug treatment, how do we move beyond what's been shown in terms of the triad and its success for chemotherapy, radiation therapy, and surgery, and move more towards the use of specific molecular targets? And now proceeding into its pathophysiology and its molecular pathology. Cancer cells have long been characterized by their phenotype histologically as visualized under the microscope. They're characterized by the distinct properties that I've already described. Cancer cells do not stop dividing when they contact a neighboring cell or the bottom of a petri plate. And they also have a greatly reduced requirement for growth factors. So these properties, along with chromosomal aberrations, have been recognized as part of the biopsy procedure and diagnosis. You can really see textbook examples, as shown here in the upper right micrograph, of abnormal cell growth and densely packed cells in a microscope slide, and you don't need to be a histologist really to recognize the abnormal appearance of these types of cells underneath a microscope. Here's a scanning electron micrograph of normal cells spreading out on a petri plate, whereas tumor cells round up into tightly balled masses, as shown here on the right. And that growth factor signal transduction activation of cell division is another hallmark of cancer cells, as I alluded to earlier, but shown again in this signal transduction pathway, which eventually results in cell division programs being activated. And again, this is a reiteration of that previous slide to remind you that there are many different characteristics that are involved when cells are liberated from their normal cell cycle. And these mutations are not necessarily communicated to offspring because they arise in somatic tissue. They are more or less confined to that organ, unless, of course, they have occurred in germline cells. So this genetic predisposition is what eventually results in tumorigenesis from a cascade effect of building mutations. Some of these are gain-of-function mutations that activate proto-oncogenes, so that activation in transcription factors which regulate promoter regions of genes can be affected. Overexpression of proto-oncogenes involved in the signal transduction pathway can also be involved, or translocations that could create hybrid proteins could also result in oncogenesis. Here's an example of some of the mechanisms of oncogene activation that could result in change of expression levels, change in protein structure that result from point mutations, or change in protein structure that could result from a hybrid protein shown in the example of the so-called Philadelphia chromosome resulting from a translocation from chromosome 9 to chromosome 22. So this disruption of cell cycle by translocation can also result in the loss of heterozygosity by mitotic recombination, where, say you had a mutant gene on one arm of a chromosome, it could be complemented by normal gene function on the other chromosome, whereas sister chromatid exchange could result in a crossing over event resulting in loss of complementation of that mutation that's involved in cell cycle control. So oncogenes then are found in signal transduction pathways, and here is a short list of some of the proteins that are involved that could become oncoproteins. I'll get back to this one here called ERBB later when we talk about Herceptin. Elimination of tumor suppressor activity requires more than one mutation where both copies of a tumor suppression gene must be inactivated in order to experience its phenotype. Remember that we have now known the human genome since 2003. Its first draft commemorated on the cover of Nature here, February 15th, 2001. And certainly this will have a large effect on our ability to diagnose as well as to develop new target drugs for cancer in the future. Knowing the human genome will also allow us to understand our predisposition to certain cancers. And in some cases, this could change lifestyles. If you have a predisposition to lung cancer, you might not want to take up smoking. On the other hand, if you have a predisposition to breast cancer, I'm not sure what you might do preventatively. Maybe more mammograms, but that has been also shown to increase the number of false positives. So, on the one hand, knowing predisposition might increase preventiveness of some cancers. On the other hand, it may increase anxiety levels and false positives, resulting in further confusion. Given the complexity of the genetics of cancer, we also have to remember that expression of a phenotype is the combination of its genetics plus its environment. And in this diagram, which I showed earlier in the pharmacogenomics section, we simply have two genes in this Venn diagram in the upper right, influenced by environment, and in the lower right, the combination of many genes, as we may see in the combination of genetics that is influencing cancer development. This is also a simplistic diagram when you consider the complexity of environmental cues, which may superimpose upon the genetic complexities. So the tools of genomics will allow us to examine predispositions that are hardwired into our inherited genetics, our ability to use transcriptomics to analyze the differences in tumor cells in their RNA populations, and that tumor cells are expressing an RNA population that is different from normal cells, and proteomics can be used to analyze the ecology of proteins resulting from the expression of those RNAs, and provide targets which we can interrupt by either small molecules or recombinant DNA drugs. So cancer is a disease of A, B, C, D, or E. We have already seen that cancers proliferate from clonal selection. Mutant cells then develop into tumor cells, which can rapidly divide. An additional mutation causes cancer cells to grow even faster and then develop into metastatic cancer. And that these cells are differentiated and have reached their final specialized form. So differentiated cells do not divide, whereas tumor cells are released from that division pathway. And that cells decide whether or not to divide at restriction points, and that these tumor suppressors can prevent passage through a restriction point. Some of these proteins will be targets specifically for certain types of drugs, such as those that are involved in tumor suppression. So mutation in DNA repair also helps keep spontaneous mutation rates low. In all cells, there are SOS repair enzymes that would recognize, say, thymidine dimers as they occur and correct for those. Certainly, mutations in DNA repair enzymes also can increase the possibility of overall mutation rates, and surveillance of those systems that protect chromosomes becomes a component of liberating those cells from their checkpoints. Cancer cells avoid senescence by inactivating tumor suppressor genes, thereby achieving the immortality that, say, was exemplified in the HeLa cell example. Telomerase is also another key player in immortalization. Telomeres, repeated DNA sequences, are found at the ends of chromosomes, which shorten from repeated rounds of cell division. Chromosomal rearrangements lead to a crisis in the ability to retain immortalization, or retain inability to go into immortalization by loss of telomere length. So, cancer is believed to be a clonal disease. The initiated events are widely thought to be, well, you can decide. And we already know that cancers are complex biosystems, and in this slide we see that cancer incidence is also a function of age. So sometimes I read that the Paleolithic diet, so-called now, eating a lot of meat and running around Central Park in bare feet, may decrease your risk of cancer because, you know, that's the way our ancestors ate. Well, truth be known, our ancestors didn't live long enough to get cancer. Most of them died prematurely of all kinds of other problems before they would develop cancer, and certainly cancer incidence increases as a function of age because the amount of time it takes to accumulate these mutations due to exposure to carcinogens is increasing over time. So there, I've increased the number of genes already linked to cancer in this slide to 490, and I think they could further be updated. But these genetic changes in DNA linked to cancer are, well, you can decide that too. This stepwise ordered genetic changes in tumorigenesis is visualized in this graph as it relates to colon tumorigenesis. And there is the progression of genes that I showed in a previous slide, where the first part of this graph shows basically when the cells are under, say, a milligram in cell number, or cell weight related to cell number. This is the dormant phase of tumor growth, whereas the later mutations correlate to rapid tumor progression phase. So hundreds of genetic events, some fertile, occur over a number of years, and it's thousands of cell divisions that are required in order for a tumor to be identifiable in a CT scan, so that sufficient time and selection pressure for the most powerful molecular advantages are to be realized by tumor cells as they go through this progression. Realize too that there is a distinct advantage to early diagnosis. We see that in the diagnostic of colon cancer through colonoscopy. So let's look at colon cancer and the promise that biotechnology will bring for its diagnosis into the future. But first, the so-called Duke staging of colon cancer was developed, first published in 1932, related to the progression histologically of colon cancer, and now recognized as Duke's classification. So colon cancer then is referring to tumorigenesis that occurs really anywhere throughout the colon, and that the staging of colon cancer relates, as it relates to Duke staging, is related to the layers of tissue in the colon wall, starting with the epithelium, where Duke's classification would recognize stage A as involving the epithelium and connective tissue of the mucosa, stage B descending down into the submucosal layers, stage C is when it has progressed into muscle tissue, and then finally stage D is all the way through the entire colon wall. And obviously stage D is the most serious progressive part of colon cancer, and the survival rate is also well correlated with surgical removal of these stages A through D. So this slide is a little funny, but it shows the progression related to mutations from normal cells to metastatic cells in cancer in the colon, and that the stages that I just showed you of Duke's progression are correlated to different mutations as they occur throughout this pathway. So the result of the clonal progression of carcinogenesis is A through E, shown here. So when we look at Duke staging of cancer, it works well only for patients stages A, B, and C, and is not very informative for predicting long-term outcomes of intermediate prognosis. The benefit of chemotherapy has seemed limited with Duke's stage C cancer. So when we look at Duke staging, it is a histological analysis, but we can imagine how molecular biology now will give us a more accurate view of biopsy and then certain treatments. So how does this correlate with molecular markers is what we really want to understand now as we move into the post-genomic era for treating not only colon cancer, but using this as an analogy for then treating other types of cancer. Going from microscope then to gene chip, companies like Affymetrix have developed cancer-specific gene chips capable of analyzing gene expression profiles that have a higher resolution than Duke's histological staging. So that we can see in this analysis of a gene chip that the gene expression pattern differs from A and B related to Duke stage B, shown on the right, versus stage C. So human cancer genes are of special interest are, and you can review that answer. Molecular staging will provide more accurate predictions of patient outcome, but certainly the use of this type of analysis will also provide a more accurate analysis of which drugs or treatments you might use. If you're unfortunate enough to develop breast cancer, but fortunate enough to develop a certain type, it will be more treatable by drugs like Herceptin coupled with tamoxifen than, say, other drugs. And we can understand this by its molecular staging versus its simply histological staging. Molecular staging of colon cancer then may have one of the following implications. So recognition of molecular staging now is moving now cancer diagnosis into a new era, rather than merely taking a biopsy and achieving its histological outcome and saying, hey, you have a breast cancer tumor. We can use molecular staging to determine exactly what type of breast cancer tumor do you have. And this may involve analysis of gene expression involved in growth factors which stimulate certain cancers. We know, for example, that estrogen and estrogen mimics play a large role in the progression of certain types of breast cancer, and that mutations that are involved in truncated receptors could cause constitutive signaling of that receptor. So in other words, if estrogen is involved in stimulation of certain types of breast or ovarian cancer, truncation of that receptor could cause on signals to continuously be released. Blocking of those receptors by various drug strategies could alleviate that symptom. Similarly, mutant RAS genes could cause those signals to always be active. And there are other types of receptor signaling, such as the TGF-beta receptor, that if it were intercepted, say by an antibody, similar to the strategy that I described for Enbrel in its treatment of rheumatoid arthritis, could be one way of interfering with a signal transduction pathway such as this, that otherwise downstream becomes extremely complicated very quickly, as I showed you in previous slides. So, molecular staging of colon cancer has implications for diagnostics of the molecular pathology, but also the interruption of cancers very early by the analysis of polyps, for example, detected during routine colonoscopies. This in itself will give rise to new biology and diagnostics. Many cancers, as we saw, are caused by field effects. A list of some of those is shown here. So, what are the contemporary target discoveries that need to be made before we can apply modern biotechnology to cancer treatments? How are those targets actually validated? When do we think, oh, disruption of a certain receptor that's involved in a signal transduction pathway might be an effective drug target? What can we use to interrupt that receptor with? Whether that's a small molecule design that could be directed, such as what we'll see for Gleevec, or antibody-based drugs, such as we might see for Herceptin. So translational medicine from important genes to important targets is at the basis now of pharmacocarcinogenomics. And the importance of gene expression towards the recognition of these receptor ligands and the multiple genes which may have a relationship to colon cancer that had been previously unknown present then the possibility of new drug targets. So to summarize part one of this lecture so far, cancer is clearly a disease of accumulating genetic changes, and that these genetic changes invariably provide key molecular advantages to those cells that enable the forming tumor to thrive and eventually to metastasize throughout the body. We are just now beginning to classify and understand tumors on the basis of these molecular and genetic changes. In part two, then, we'll look at approaches to developing new drugs that take away some of the advantages of targeting molecular changes itself, but also limiting the vast side effects that people suffer from traditional chemotherapy and radiation therapy. What we really want to do is specifically target the molecular structures that are specifically involved in cancer cell biology.